Welcome to Fish Geekery. This week we'll be looking at some research from the Woods Hole Oceanographic Institution in Woods Hole, Massachusetts. This article was originally published in January 2012 in the magazine Oceanus. You can find a citation and a link to the article in the description below. Woods Hole and MIT Joint Program student Li Ling Hamady wanted to fill in some of the blanks in humanity's big book of scientific knowledge. His focus was on the basking shark. Basking sharks are the world's second largest fish after whale sharks. Their fins are highly valued as a delicacy food, and with levels of protection changing as they swim through international waters, they've been overfished for generations. Like many other large animals, they're slow-growing, long-lived, slow to mature, and have low fertility, making them vulnerable to continued fishing pressure, whether they're caught intentionally or accidentally. The basking shark has been listed as vulnerable to endangered by the International Union for the Conservation of Nature, or IUCN. All species listed by the IUCN need protection, but the basking shark presents a problem. Half of the time, we don't know where they are. The sharks are generally seen swimming slowly near the surface, with their huge mouths open, filtering out plankton to eat. Most observations are made in the relative shallows near the edges of the continents, in both the northern and southern hemispheres. The problem is, these observations are only made for about half of the year. So, where do they go for the rest of the time? Since humans are land-dwelling air breathers, we can't follow the sharks into the depths, and chasing them with boats and submarines would probably change their habits anyway. I know my habits would change if I were being stalked by a bunch of people with clipboards. Since we can't watch them ourselves, the next best thing is tagging. The tags used here are electronic devices that record depth, light level, and water temperature. Researchers attached them just below the dorsal fin using a harpoon. I know, ow! But in the same way that it's generally better to get a shot from your doctor than it is to die of some horrible disease, getting tagged so that scientists can learn enough to effectively protect you and your friends is generally better than being hunted into extinction. The tags stay on the sharks for a pre-programmed length of time, up to a year, then detach and return to the surface. Once they're there, they transmit the data that they've been gathering back to the researchers. Since it's difficult to pinpoint locations in the ocean, the GPS in your car uses satellite signals which get messed up when they try to go underwater, scientists figure out where the sharks have been based on the growth rings in their vertebrae. The vertebrae came from sharks who had stranded on beaches or had died out at sea and whose bodies had washed up on shore. In the same way that trees lay down new growth rings every year, basking sharks form distinct layers in the cartilage of their vertebrae. Since at first nobody knew how often these layers formed, Hamady had to find some known reference point and work from there. One method of figuring out how old something is by measuring how much of the radioactive isotope carbon-14 is present. Every living thing, including you and I, have carbon-14. Since it's unstable, however, when we die, it begins to decay into the more stable carbon-12. We're not going to get into the specifics of that right now because, frankly, I'm still trying to figure out chemistry myself, and it just wouldn't feel right trying to teach it to you when I don't fully understand it, but I'm sure there are plenty of chemists out there who would be more than happy to answer any questions you have. We know how quickly this decay happens, so figuring out when something stopped living is a relatively straightforward process. In this case, we're not measuring when the shark died, but when the plankton that the shark ate to give it the nutrients to form that layer of cartilage died. Today's lunch died today, last week's lunch died last week, that kind of thing. While this kind of carbon dating is great for archaeological finds, it isn't precise enough to measure seasonal changes. That's where that known reference point comes in. With the development, testing, and use of nuclear weapons in the 1940s and 50s, there was a sudden spike in the amount of radioactivity in the atmosphere, which then mixed down into the oceans. These levels started dropping off again with the Limited Test Ban Treaty of 1963. Since basking sharks are so long-lived, many of the ones used in this study had growth layers that were formed while carbon-14 levels were still relatively high. Hamady compared the amount of carbon-14 in the growth layers to known levels of radiation in the environment. From there, he could figure out seasonal differences within each year of growth. So how exactly does this help us find where the basking sharks go? The ocean isn't the same everywhere you go. Different regions have different levels of carbon and nitrogen, and by finding the ratio of those two in each layer of cartilage, a comparison to a map of what scientists call the isotope landscape, or isoscape, reveals where the shark was feeding. Together, these data have showed that a group of basking sharks who spend summers off of Cape Cod travel to the warmer waters of the Caribbean Sea and even across the equator into Brazil when the waters off New England get a bit too chilly. With this new knowledge, we can finally start working on an effective international conservation plan that will help protect the basking shark. And, through the techniques developed in this study, other migrating marine species as well. Thanks for joining us this week on Fish Geekery. If you have any questions, comments, or suggestions, you can reach us on Facebook, Twitter, or of course in the comments below.